Bismillahirrahmanirrahim. Assalamualaikum and a very good morning, everyone. Can you hear me?
Yes, Doctor.
Okay, thank you. Shall I start?
Yes, Dr. Rosina.
Thank you. So today we're going to learn about the electrical properties of cells and also membrane potential. So in this lecture, what you need to know is about resting membrane potential, okay, action potential, and graded potential, and also what are the difference between action potential in the neuron and also in cardiomyocyte. Okay. I beg your pardon? Anything?
No, can continue.
Okay, thank you. So these are the things that you need to know. So basically, this is just a very simple and easy lecture. At the end of my session tomorrow, right, I'll give you a case scenario questions for all the system in physiology. Okay, it's a questions with case scenario together with the questions, like a short notes question and also the one best answer question together with the brief with the explanation of the answers. So I'll give it to you tomorrow so that you can have time to revise before your exam. Okay. So in this lecture, at the end of this lecture, you need to know, you must be able to describe the cell properties in cell in a resting membrane potential. Okay. What's inside the cells? What are the factors contributing to unequal distribution of membrane potential? Okay. You need to understand the terms used in membrane potential. You need to describe the process in action potential and graded potential in a cell. Okay. Compare and contrast the action and graded potential. Okay. In action potential, you have absolute and relative refractory period. So you need to know how to distinguish between these two. Okay. Absolute and relative refractory period. Okay. And then describe the action potential in cardiac muscle, cardiomyocytes. Okay, compare and contrast the action potential in the cells, neuron, and also in cardiomyocytes. Okay, and what is the physiological functions of action potential? So for example, in order for you to understand my lecture, right, the action potential is going on, okay? So you need the action potential so that the information will be transmitted to your brain and for you to understand. Okay, so cell properties. In a resting cells, right, there is a potential difference. That means due to different amount of charges across the plasma membrane. So inside of the cells normally negatively charged, outside of the cells positively charged. Okay, so when there is no action potential, it's called the membrane has a resting membrane potential. Okay, in the cells, in the nerve, it's about -70 millivolt. In cardiomyocyte, the resting membrane potential is about -90 millivolt, okay, before an action potential can be generated. Okay, so inside is negative relative to outside. So when there is a positive charge enters the cells might exhibit membrane potential. So that is called polarized. Okay, so that is where the membrane potential from -70 millivolt increase to more positive. Okay, so muscle fibers, neuron are electrically excitable. So they communicate with one another using two types of signals. One is action potential, another one is graded potential. So action potential allows communication over both short and long distance within the body. Okay, graded potential normally used for short distance communication only. Okay, production of these two types of signals depend on basic features of the plasma membrane. So The first one is the existence of the resting membrane potential, okay, and then the presence of specific ion channels. So you must have specific ion channels in order for the membrane potential to occur. For example, in a normal cells, neurons, you need sodium, potassium channels, okay, and then for cardiomyocytes, you need sodium fast channels, potassium, and also calcium channels. Okay, the resting membrane potential, RMP, is maintained by two factors. Okay, the first one is unequal distribution of ion across the plasma membrane. So the extracellular fluid is rich in sodium and chloride. The intracellular fluid is rich with potassium, phosphate, and proteins. Okay, relative permeability of plasma membrane to sodium and potassium, that is also important. In a resting membrane neuron or muscle fiber, the permeability of plasma membrane is 50 to 100 times greater to potassium rather than sodium to sodium ion. Okay. So now let's go through the terms in graded and action potential. Okay. So what is depolarized? Okay. When depolarized, that means when there's positive ion like sodium entering into the cells, so it causes more positive charge inside of the cells, which previously the inside of cells is more negative rather than on the outside of the cells. So when the positive charge enter, that is called depolarizing. So depolarized potential is less negative than the resting membrane potential. So resting membrane potential, I've already mentioned, in a muscle cells, neuron is negative 70 millivolt. In cardiomyocytes is negative 90 millivolts. Okay, then overshoot. What is overshoot? Is a reversible membrane polarity. That means more positive ion, sodium ion enters into the cells. The more sodium ion enters, the more the charges of the membrane potential will become more positive. So sometimes it lead to supra threshold level. Okay, so that is called overshoot. Okay, repolarizing, that is where the sodium ion channels closed. That's where the potassium ion flow outward. Okay, so that is called repolarizing. So the membrane potential that have been depolarized, return to the resting values. Okay, hyperpolarized is the potential more negative than the resting level. Okay, we will see in which condition that it causes hyperpolarized, repolarized, depolarized, and overshoot. Okay, we'll see after this. Okay, there is also another term that you need to understand, the inward current, flow of positive charge into the cells, and outward currents, flow of positive charge out of the cells. Threshold potential, MP here is membrane potential. Okay, please take note. Membrane potential causing the occurrence of action potential. Okay, refractory period is a period during another action potential cannot be elicited in the excitable tissue. So first you need to define what is refractory period. In refractory period, you have absolute and relative refractory period. Okay, there's two differences. So it's a period during another action potential cannot be elicited. Okay. So now let's go to the graded potential. What is graded potential? When a stimulus causes a ligand-gated or mechanical gated ion channels to open and close, okay, in an excitable cell's plasma membrane, so it produces a graded potential. So graded potential makes the membrane either become more polarized or less polarized, okay? So it can be depolarized or hyperpolarized, less polarized, okay? So when a response is more negative polarization, then it causes hyperpolarization graded potential, okay? And when the response is less negative polarization, that means it's caused depolarization, okay? So ligand-gated, mechanically gated ion channels present at the dendrite and also the sensory neurons. Okay, flow of the current is localized, spread along the membrane only for a few micrometers before it dies out. So it's for short distance communication only, okay, for graded potential. Action potential, it goes long and long, okay? Okay, so graded potential, you can find it in dendrites, cell body, okay? And this is the hyperpolarization, that means more negative, and this is the depolarization graded potential, okay? Still under graded potential, okay? Ligand gated, you can find it most numerous in the dendrites, cell body of the interneurons, motor neurons, and also in axon. Okay, examples are receptor potential, postsynaptic potential, and pacemaker potential. Okay, so graded potential is used for short distance communication. Okay, so this is the depolarization and hyperpolarization. Right, now we go for action potential. What is action potential? So it's a phenomena of excitable tissue, nerve, and muscles that occurs in nerve and also muscles. It's for long distance signal, okay? In order for you to understand better the action potential, you will learn about the neuromuscular contraction, right? So in order for you to have muscle contraction, you need to have action potential first. This is quite a basic lecture, okay? So that's why I say action potential. In order for you to understand my lecture, the action potential is going on, okay? So long distance signal, okay? It's for long distance signal. So it's a voltage gated channel that gives membrane the ability to undergo the action potential. It occurs very rapidly within 1 to 4 milliseconds, okay? Repeat at frequencies of several hundreds per second, okay? In terms of stereotypical and size and shape, it depolarized and repolarized and back to the same resting membrane potential. So it has the same shape. Depolarized and repolarized and back to resting membrane potential. Okay, it propagates. Action potential at one site causes depolarization to adjacent sites, okay, non-decremental. All or non-response is a law whether if the stimulus is strong enough, action potential will occur. If the stimulus is not strong enough, okay, it doesn't reach a certain voltage, then action potential could not occur. So that's what it meant by all or non-response. Action potential occur or not occur, okay, depending on the voltage stimulus. Okay, so far, do you understand?
Yes.
Yes.
Thank you. Okay, so now we go for the action potential mechanism. Okay, as I mentioned just now, you have the depolarization and repolarization. So now we will see where is the depolarization curve and repolarization curve. Okay, if you see here at the resting state, all voltage gated channels, sodium channels are closed. Okay, so this one here, negative 70 millivolts is the resting membrane potential. Okay, so where all the activated channels, sodium channels and potassium channels are closed. Okay, in depolarization phase, right? When the sodium channels open, okay, sodium inflow into the cells. Okay, what happened inside of cells is more negative. So when positively charged sodium enters to the cells, it becomes more positive, right? So it becomes depolarized. So inflow of the sodium will further depolarize the membrane, okay? So opening the more sodium channels activation gate. So as you see, when the sodium flow into the cells, the voltage from -70 right, to more positive, you call depolarization. Okay, this green line here is depolarization. Okay, at some point, okay, once it reached its threshold, right, the sodium channels were closed. Okay, the sodium channels will be closed, activation gates will close, and that's where the potassium ion will outward flow of the potassium, causing the repolarization. Causing the repolarization. Outflow of the potassium ion, okay, from the cells, okay, will cause repolarization. Will cause repolarization. That's where the red curve here. So potassium outflow restores the resting membrane potential. So it will outflow until it reach the resting membrane potential. Okay. So return to the resting gate when the potassium gate is closed. Okay. So potassium will continue outflow when it returns to the resting membrane potential, then it closed. Okay. So this is how the action potential mechanism works. Okay. So this is the resting membrane potential. This is where you have the sodium conductance when the sodium enters into the cells. Okay. At some point, at 30 millivolt voltage, that's where the sodium channel closed. Okay. And then no more sodium going into the cells. Okay. And that's where you have potassium conductance. So potassium will be outflow from the cells to maintain the what? The inside more negative and more positive outside of the cells. Okay, and then the sodium conductance potassium will continue to outflow from the cells until it reach the resting membrane potential. Okay, so in action potential, these two gates that is very important. Okay, okay, what is refractional period or refractory period? Okay. The period of action potential which of an excitable cells cannot generate another action potential. That is called refractory period. So during refractory period, you cannot generate another action potential. So, but there's two conditions. There are two conditions where you have absolute and relative refractory period. Okay, during absolute refractory period, a second action potential cannot be initiated, even though you have a very supra-threshold stimulus. Okay, even you have very strong stimulus, you still cannot stimulate another action potential. Okay, but in the relative refractory period, you can generate another secondary action potential, but with a supra-threshold stimulus. Okay, let's see where is the absolute and relative refractory period. Okay, so if you see here, during depolarization and polarization, this period is. Okay, so that's why you cannot stimulate another action potential even though you have a supra-threshold stimulus. Okay, because you have an action potential here. But if you see here, this this is the relative refractory period. Okay, where it's already reached to the the outflow of the potassium have made the membrane voltage, membrane potential gone to the resting membrane potential. Here you can stimulate another action potential as long you must reach a supra-threshold stimulus. So you can stimulate another action potential here, but with a supra-threshold stimulus. So this is called relative refractory period. So remember absolute and relative refractory period. Okay. Okay. What are the functions, physiological functions of action potential? So the first one is transmission of nerve impulse, contractions of muscles, activate and inhibit glandular secretion, release of chemical transmitter in the synapse. Okay, so normally for undergraduate level, I normally ask them how to compare and contrast the graded and action potential. So this is used to answer their marking slip for them. So you need to as a postgraduate level, this is a revision. You must know now what is graded potential, action potential. Okay, where does it come from? The origins. Okay, graded potential from the dendrites, cell body. Okay, action potential triggers zones and propagate along the axon. Okay, type of channels. Graded potential is ligand-gated and mechanically gated ion channels. Action potential is more on the voltage-gated ion channels. Okay, proper conductions, graded channels does not propagate. Action potential propagates, and that's why it's communicating for over long distance. Okay, so the amplitude is less than 1 millivolt to 50 millivolt. And here, all or non-response, whether it depends on the stimulus. If the stimulus is strong, then action potential occurs. If it's not strong, then it'll be like no action potential. So typically it's about okay. And then this one longer ranging, several milliseconds to several minutes. This one shorter ranging, okay, because that's why it's moving faster, okay. And this one polarity may be hyperpolarized, okay, or depolarized. And this one always you have the same curve, depolarized and repolarized, and then back to resting membrane potential and back to action potential, depolarized and repolarized back. That's the same pattern. So refractive period don't have, so it have a spatial and temporal summation, which you will learn this in the neuromuscular contraction. And then this one here, action potential do not exhibit summation. Okay. Okay, now before we go to the action potential in cardiomyocytes, do you have any questions in resting membrane potential, action potential, and gradient potential? Do you understand?
Yes. Like.
Okay, now let's go to the action potential in cardiac muscles. Okay, so there are five phases, okay, phase 0 to phase 4, okay. So we start with the phase 4, the resting phase. So as I mentioned just now, normal cells, the neurons, okay, they have the resting membrane potential is negative 70 millivolt. But for cardiomyocytes, it's negative 90 millivolt, okay? Okay, so the resting membrane potential in cardiomyocytes is 90 millivolt due to a constant outward leak of potassium through the inward rectifier channels. Okay, I'll show you the graphs after this, okay? So sodium channels and calcium channels are closed at the resting transmembrane potential. So TMP here stands for transmembrane potential. So action potential in normal cells involve sodium and potassium channels. But in cardiomyocytes, there are 3 channels, sodium fast channels, potassium, and also calcium channels, okay? So this is what I meant, the 5 phase. So we start with phase 4, okay? Sodium and calcium channel closed. So there is opening of potassium channels just to rectify the channels to create the transmembrane potential stables at negative 90 millivolt. So now we go at phase 0, okay, where you have rapid influx of sodium fast channels. Okay, so now we see. So phase 0 is called as depolarization. Okay, depolarization. So action potential triggered in the neighboring cardiomyocyte or pacemaker cells, causing the transmembrane potential to rise above negative 90 millivolts. Okay. So action potential is triggered in the neighboring, so fast channels start to open one by one. Fast channels start to open one by one, and the sodium leaks into the cells. So when the sodium leaks into the cells, right? When the cells inside of cells is negatively charged, right? So when sodium enters in, it becomes more positive. So still under depolarization, okay, the transmembrane potential approaches negative 70 millivolt. So it will allow more fast sodium channels, more sodium ion to enter into the cells. So the large sodium ion current depolarize the transmembrane potential to 0 millivolt and slightly above 0 millivolt, okay? So that is called the period where you call overshoot. Remember when I explained the term of overshoot, you have the term of overshoot. So it's slightly higher than the 0 millivolt, okay? So when it's rising up to more than 0 millivolt, right? When it reaches the negative 40 millivolt, the calcium channels also started to open already, okay? The long tight calcium channels open when the transmembrane potential is greater than negative 40 millivolt. Okay, during depolarization, once it reached negative 40 millivolt, that's where the long type calcium channels open. Okay, so it causes a small but steady influx of the calcium down to its concentration gradient. Okay, So this is what happened, okay? So when it's negative 40 millivolt, okay, so it goes rise 1, okay, phase 1, early repolarization. So what happened in early repolarization? Now the transmembrane potential is now slightly positive. Okay, some of the potassium channels open, so there is outflow of the potassium channels. Potassium ion returns the transmembrane potential to 0. Okay, so when there's opening of potassium channels, right, outflow of the potassium from the cells, right? So let's say this one here at 20 millivolt, okay? So now I have returned to 0. So there's opening of the potassium channels that makes the transmembrane potential return to 0 millivolt here, okay? So now we go to phase 2. Phase 2, this one here is the plateau phase. Okay, phase 1 here, plateau phase. Phase 2, plateau phase. The L-type calcium channels are still open, okay? Still open, and there is a small constant inward current of calcium. Okay, this becomes significant in the excitation contraction coupling process. Okay, so potassium leak down to its concentration gradient. Currents are electrically balanced. So the TMP is maintained at plateau, just below 0 millivolt throughout the phase 2. So during phase 2, there is contraction of the cardiomyocytes. Okay. So during plateau phase, there is a contraction, okay, contraction. And now we go to phase 3. Okay, phase 3, repolarization. So now in phase 3, the calcium channels are gradually inactivated. Okay, so there is persistent outflow of the potassium ion, okay, now exceeding the calcium inflow. Okay, so it brings back the transmembrane potential to resting membrane potential, which is negative 90 millivolt. Okay, to prepare the cells for the new cycle of depolarization. Okay, so membrane and calcium ion to the extracellular environment and the potassium to the cell interior. Okay, so the pump involved include the sarcolemma sodium calcium exchanger, calcium ATPase, and sodium potassium ATPase pump. So all these pumps are responsible to bring back the normal transmembrane ionic concentration once it has reached the repolarization. Okay, so where is the absolute refractory period? Where do you think the absolute refractory period? Anybody? The 1 here until the 2, okay? So this is the early refractory period, okay? So during this period, you have depolarization, repolarization. So this is the absolute refractory period. So you cannot have contraction. You have actual contraction here, okay? This is so this is the depolarization, repolarization. So this is between here, okay, the phase 1 here until here, okay, until but this refractory period. Why it has a long refractory period? Because contraction occurs, and you need to allow relaxation of the cardiac muscles, cardiomyocytes. Okay, so there is contraction and relaxation before the next action potential for the, again, the same process of depolarization and repolarization to occur. Okay, so this is the refractory period, okay, and this is the contraction, and you have relaxation. So that's why in cardiomyocytes, it has a longer refractory period because compared to other cells, because to allow the contraction and also relaxation of the cardiomyocytes. Okay. So this is again, you see here, the sodium conductance is higher during the phase 1, okay? And then the calcium during the plateau phase, the conduction of the calcium ion. And then the conductance of the potassium ion is higher during the repolarization phase, okay? So depolarization, repolarization, and then you have 200 milliseconds for contraction and the balance remaining for the relaxation. Okay, so that's why in cardiomyocytes, you have a longer refractory period to allow contraction and relaxation. Okay. Is it possible to watch the YouTube? Just open it.
Stuffy smell at home? It's odors and germs trapped in fabrics you can't wash daily. Spray Febreze. It removes odors and kills up to 99.9% of viruses and germs.
First, I think you have to share your screen.
Just hold on, just hold on. Okay.
The heart is essentially a muscle that contracts and pumps blood. It consists of specialized muscle cells called cardiac myocytes.
Yes, okay.
Action of these cells is initiated by electrical impulses known as action potentials. Unlike skeletal muscles, which have to be stimulated by the nervous system, the heart generates its own electrical stimulation. In fact, the heart can keep on beating even when taken out of the body. The nervous system can make the heartbeats go faster or slower, but cannot generate them. A small group of myocytes called the pacemaker cells, which constitute the cardiac conduction system. These are modified myocytes that lose the ability to contract and become specialized for initiating and conducting action potentials. The SA node is the primary pacemaker of the heart. It initiates all heartbeats and controls heart rate. If the SA node is damaged, other parts of the conduction system may take over this role. The cells of the SA node fire spontaneously, generating action potentials that spread through the contractile myocytes of the atrium. The myocytes are connected by gap junctions, which form channels that allow ions to flow from one cell to another. This enables electrical coupling of neighboring cells. An action potential in one cell triggers another action potential in its neighbor, and the signals propagate rapidly. The impulses reach the AV node, slow down a little to allow the atria to contract, then follow the conduction pathway and spread through the ventricular myocytes. Action potential generation and conduction are essential for all myocytes to act in synchrony. Pacemaker cells and contractile myocytes exhibit different forms of action potentials. Cells are polarized, meaning there is voltage across the cell membrane. In a resting cell, the membrane voltage, known as the resting membrane potential, is usually negative. This means the cell is more negative on the inside. At this resting state, there are concentration gradients of several ions across the cell membrane. More sodium and calcium outside the cell, and more potassium inside the cell. These gradients are maintained by several pumps that bring sodium and calcium out and potassium in. An action potential is essentially a brief reversal of electric polarity of the cell membrane and is produced by voltage-gated ion channels. These channels are passageways for ions in and out of the cell, and as their names suggest, are regulated by membrane voltage. They open at some values of membrane potential and close at others. When membrane voltage increases and becomes less negative, the cell is less polarized and is said to be depolarized. Reversely, when membrane potential becomes more negative, the cell is repolarized. generated, the membrane voltage must be polarized to a critical value called the threshold. The pacemaker cells of the SA node spontaneously fire about 80 action potentials per minute, each of which sets off a heartbeat, resulting in an average heart rate of 80 beats per minute. Pacemaker cells do not have a true resting potential. The voltage starts at about -60 millivolts and spontaneously moves upward until it reaches the threshold of -40 millivolts. This is due to action of so-called funny currents present only in pacemaker cells. Funny channels open when membrane voltage becomes lower than -40 millivolts and allow slow influx of sodium. The resulting depolarization is known as pacemaker potential. At threshold, calcium channels open. Calcium ions flow into the cell, further depolarizing the membrane. This results in the rising phase of the action potential. At the peak of depolarization, potassium channels open, calcium channels inactivate, potassium ions leave the cell, and the voltage returns to -60 millivolts. This corresponds to the falling phase of the action potential. The original ionic gradients are restored thanks to several ionic pumps, and the cycle starts over. Electrical impulses from the SA node spread through the conduction system into the contractile myocytes. These myocytes have a different set of ion channels. In addition, their sarcoplasmic reticulum, the SR, stores a large amount of calcium. They also contain myofibrils. The contractile cells have a stable resting potential of -90 millivolts and depolarize only when stimulated, usually by a neighboring myocyte. When a cell is depolarized, it has more sodium and calcium inside the cell. These positive ions leak through the gap junctions to the adjacent cell and bring the membrane voltage of this cell up to the threshold of -70 millivolts. At threshold, fast sodium channels open, creating a rapid sodium influx and a sharp rise in voltage. This is the depolarizing phase. L-type or slow calcium channels also open at -40 millivolts, causing a slow but steady influx. As the action potential nears its peak, sodium channels close quickly, voltage-gated potassium channels open, and these result in a small decrease in membrane potential, known as early repolarization phase. The calcium channels, however, remain open, and the potassium efflux is eventually balanced by the calcium influx. This keeps the membrane potential relatively stable for about 200 milliseconds, resulting in the plateau phase, characteristic of cardiac action potentials. Calcium is crucial in coupling electrical excitation to physical muscle contraction. The influx of calcium from the extracellular fluid, however, is not enough to induce contraction. Instead, it triggers a much greater calcium release from the SR in a process known as calcium-induced calcium release. Calcium then sets off muscle contraction by the same sliding filament mechanism described for skeletal muscle. The contraction starts about halfway through the plateau phase and lasts till the end of this phase. As calcium channels slowly close, potassium efflux predominates and membrane voltage returns to its resting value. Calcium is actively transported out of the cell and also back to the SR. The sodium-potassium pump then restores the ionic balance across the membrane. Because of the plateau phase, cardiac muscle stays contracted longer than skeletal muscle. This is necessary for expulsion of blood from the heart chambers. The absolute refractory period is also much longer, 250 milliseconds compared to 1 millisecond in skeletal muscle. This long refractory period is to make sure the muscle has relaxed before it can respond to a new stimulus and is essential in preventing summation and tetanus, which would stop the heart from beating. Thank you for watching. Please feel free to suggest a topic.
Let me tell you a story about the time when I first graduated, broke and jobless, and my father scolded me.
Neurons communicate with each other through their dendrites and axon. Generally, incoming signals are received at dendrites, while outgoing signals travel along the axon to the nerve terminal. In order to achieve rapid communication over its long axon, the neuron sends electrical signals from the cell's body to the nerve terminal along the axon. These are known as nerve impulses or action potentials. An action potential is essentially a brief reversal of electric polarity across the cell membrane. Cells are polarized, meaning there is an electrical voltage across the cell membrane. In a resting neuron, the typical voltage, known as the resting membrane potential, is about -70 millivolts. The negative value means the cell is more negative on the inside. At this resting state, there are concentration gradients of sodium and potassium across the cell membrane, more sodium outside the cell and more potassium inside the cell. These gradients are maintained by the sodium-potassium pump, which constantly brings potassium in and pumps sodium out of the cell. A neuron is typically stimulated at dendrites, and the signals spread through the soma. Excitatory signals at dendrites open ligand-gated sodium channels and allow sodium to flow into the cell. This neutralizes some of the negative charge inside the cell and makes the membrane voltage less negative. This is known as depolarization, as the cell membrane becomes less polarized. The influx of sodium diffuses inside the neuron and produces a current that travels towards the axon hillock. If the summation of all input signals is excitatory and is strong enough when it reaches the axon hillock, an action potential is generated and travels down the axon to the nerve terminal. The axon hillock is also known as the cell's trigger zone, as this is where action potentials usually start. This is because action potentials are produced by voltage-gated ion channels that are most concentrated at the axon hillock. Voltage-gated ion channels are passageways for ions in and out of the cell, and as their name suggests, are regulated by membrane voltage. They open at some values of the membrane potential and close at others. For an action potential to be generated, the signal must be strong enough to bring the membrane voltage to a critical value called the threshold, typically about -55 millivolts. This is the minimum required to open voltage-gated ion channels. At threshold, sodium channels open quickly. Potassium channels also open, but do so more slowly. The initial effect is therefore due to sodium influx. As sodium ions rush into the cell, the inside of the cell becomes more positive, and this further depolarizes the cell membrane. The increasing voltage in turn causes even more sodium channels to open. This positive feedback continues until all the sodium channels are open and corresponds to the rising phase of the action potential. Note that the polarity across the cell membrane is now reversed. As the action potential nears its peak, sodium channels begin to close. By this time, the slow potassium channels are fully open. Potassium ions rush out of the cell, and the voltage quickly returns to its original resting value. This corresponds to the falling phase of the action potential. Note that sodium and potassium have now switched places across the membrane. As the potassium gates are also slow to close, potassium continues to leave the cell a little longer, resulting in a negative overshoot called hyperpolarization. The resting membrane potential is then slowly restored thanks to diffusion and the sodium-potassium pump. During and shortly after an action potential is generated, it is impossible or very difficult to stimulate that part of the membrane to fire again. This is known as the refractory period. The refractory period is divided into absolute refractory and relative refractory. The absolute refractory period lasts from the start of an action potential to the point the voltage first returns to the resting membrane value. During this time, the sodium channels are open and subsequently inactivated while closing and thus unable to respond to any new stimulation. The relative refractory period lasts until the end of hyperpolarization. During this time, some of the potassium channels are still open, making it difficult for the membrane to depolarize, and a much stronger signal is required to induce a new response. During an action potential, the sodium influx at a point on the axon spreads along the axon, depolarizing the adjacent patch of the membrane, generating a similar action potential in it. The sodium currents diffuse in both directions on the axon, but the refractory properties of ion channels ensure that action potential propagates only in one direction. This is because only the unfired patch of the axon can respond with an action potential. The part that has just fired is unresponsive until the action potential is safely out of range. An action potential generated at the axon hillock usually travels down the axon to the nerve terminal and not back to the cell body. This is because the concentrations of voltage-gated ion channels are higher in the axon than in the cell body.
Okay, I hope you understand my lecture. Do we have more time to ask questions? I have some sample of MCQ questions, or if you don't have time, then I can email to.
Sweet Jesus and Cajuns. Get the BTS meal at McDonald's.
So can I just shoot another one or two questions? Can I? I have this time some more, right? 15 minutes. Fatin, is that okay?
Of course, Dr. Roslina.
Okay, thank you. So I'm just going to read the question and you answer, you tell me which are the answer, correct answer, okay? Okay, regarding depolarization. So A, it is associated with increase in membrane permeability to sodium ion. B, it's terminated with closure of voltage activated potassium channels. C is followed by muscle relaxation. D is caused by potassium efflux.
A.
Right, the answer is A. It's associated with increased membrane permeability to sodium ion. Good. Okay, the second question regarding action potential. The first A is graded potential. B is produced by subthreshold stimulus. C, it starts with repolarization, cause outward movement of chloride ion. D is conducted slowly in thin nerve fibers. You want me to repeat the question?
Repeat the question, Doctor.
Okay. Regarding action potential. Action potential is a graded potential. B, action potential is produced by subthreshold stimulus. C, starts with repolarization caused by outward movement of chloride ion. D is conducted slower in thin nerve fibers.
D.
Boy?
Some answer boy, some answer donkey.
The answer is donkey. It's conducted slower in thin nerve fibers. Okay, so don't just depend on my lecture notes, but read also some of the textbook. Okay. So this one I did not mention in the lecture notes just now. Okay. Can I have one more question?
Dr. Rosani, can I ask a question? This is Haris from Surgical.
Yeah.
Hello, Dr. Rosani.
Yeah.
Dr. Rosina.
Yeah.
I want to ask regarding the action potential in the small, in the bowel, because the book mentioned about the gastric slow wave. What is it, and what is the difference between the action potential in the muscle, in the normal muscle, and what is the significance of it?
Action potential. Can you repeat back your question?
I want to ask regarding the action potential in the bowel. peristalsis to occur, because the book the book mentioned about the gastric slow wave. The term is a gastric slow wave, but I'm not sure what is the significant and what makes it different from the other action potential. It's
gastric moving slowly, that is the peristalsis, right?
yes, because they have some diagram shows like action potential, but they said this is not true action potential for the bowel.
Can I double check and get to you back? Can you WhatsApp me your questions? Then I email back.
Okay.
Is it possible? Thank you.
Okay.
I need to clarify some things. Okay, I have another question. Okay, the resting membrane potential is caused by A, diffusion of potassium ion outside the nerve fibers, B, diffusion of sodium ion inside the nerve fibers, C, opening of the chemical activated ion channels, D, opening of voltage activated ion channels. You want me to repeat the question?
Yes.
Repeat again.
Okay, the resting membrane potential is caused by A, diffusion of potassium ion outside the nerve fibers, B, diffusion of sodium ion inside the nerve fibers, C, opening of the chemically activated ion channels, D, opening of voltage activated ion channels. Donkey, yeah? Yes. The resting membrane potential here, not action potential. The resting membrane potential.
Yes.
I'm not asking about action potential. The resting membrane potential.
It's a B, yeah.
Are you sure it's D?
A.
What happened before the action potential occur? If the membrane, inside the membrane there is
ion efflux.
which ion influx? Which one do you think, A or B? A or B?
A.
The answer is A. So when you read the question, read carefully. Is it action potential or resting membrane potential or graded potential? There's three different things, you know. So sometimes if you read faster, you never read properly, you can get the wrong answer. So if you think the resting membrane potential, you straight away think about action potential. So you will answer D. So the answer is wrong. So you read back the clause, you say the resting membrane potential is caused by. So when the cells is more positive inside, so of course the sodium potassium ATPase pump will correct it, right? So there is a diffusion in potassium ion outside the nerve fibers. Okay, just to maintain the resting membrane potential. Okay, you understand?
Yes.
So when you read the question, you read carefully. Okay, for tomorrow, I have respiratory physiology with you guys. Okay, I need to ask you, for respiratory physiology, there are 6 lectures when I teach undergrad. So there is mechanics of breathing, exchange of gases, ventilation, perfusion, regulation of respiration, and then hypoxia and acclimatization to higher and lower altitude. Out of these 6 lectures in 1 hour, which one do you want me to focus more? Maybe you can discuss later and ask your class rep to text me. Is it okay?
Okay, Doctor.
Okay, so there are 6 lectures. So if you can tell me which one to focus on, then maybe I can focus on that. But I'll give you all the 6 lecture notes. So I'll give you everything, but during the 1 hour lecture, I'll focus more on what you request. Is that okay?
Okay, can.
Okay, thank you.